Well, thank you very much for the kind introduction, and I'd like to thank the meeting organizers for inviting me to speak here today. And yeah, I'd like to, is that better? I'd like to thank, no. Is this on? Ah, okay. What can I do here? Is that better? Is that better? Yes? No. Putting it in. Better? Is that better? No. So, is it this microphone that's? Okay. Is this better? Okay. So I'm just going to have to lean in. Gee, I'm usually not worried about my voice carrying. My kids had no trouble hearing my voice and selectively tuning it out as they were growing up. I'd like to thank the meeting organizers for inviting me to speak here today and commend them on this very comprehensive view of GM crop development, starting all the way with the development of the crops themselves. So this presentation today will focus on how biotechnology fits into the overall picture of crop genetic improvement and the development of new cultivars. And specifically, we'll look at the biotech trait development pipeline and details of event selection. And it will look at the pipeline for trait introgression, that is to move the transgenic event into newly developed improved cultivars. And then finally, we'll briefly touch on quality control elements for monitoring commercial seed that goes out into the marketplace. So this presentation deals with two product pipelines and the data that are generated in order to make commercialization advancement decisions through the process. It does not focus, for example, on data that are collected specifically for the regulatory submission package, which Laura Preval will touch on in the next presentation. So plant breeding is the science of applying genetic principles to improve plants for human use. And basically, plant breeding impacts the life of every person on the planet. Why? Because it involves the creation and manipulation of economically important properties in plants that are used for food, feed, fuel, fiber, and flora. Plant breeding's been enormously successful. I'm going to be giving examples today mainly from corn because that's the crop I work with, and it's also the crop in which we probably have the most experience with transgenic product development. Plant breeding's been enormously successful in corn. So looking back to the 1930s and open pollinated varieties used in the U.S., the average yield was about 30 bushels per acre. And in 2000, the average yield for corn in the U.S. with hybrids was 150 bushels per acre. So five-fold yield increase over the course of 70 years. And then, as breeders look forward to the future, we're looking to, again, in corn, double yields compared to the year 2000. So we're aiming for 300 bushel per acre by the year 2050, as are nearly all plant breeders across different crops trying to double or triple the yields of their crops by the year 2050. So we're all working very hard to feed a growing world population, one that's growing by 73 million per year and expected to reach 9 billion by the year 2050. Also a world population that's consuming more meat, and so grain demands are greater, especially in emerging economies. At the same time, we don't expect any new appreciable land to be devoted to agriculture. Water tables around the world are falling, and climate change and more extreme weather patterns are introducing a greater element of risk in crop production and the possibility for food insecurity. So the concept of plant breeding is actually very simple. Cross the best parents and identify the most outstanding progeny that outperform the parents. So what I'm showing here is the cycle of creating and exploiting genetic variation. So when we cross those best parents, plant breeders are creating useful genetic variation in a breeding population from which they will make selections among progeny to exploit that genetic variation toward very specific breeding objectives. So in the 21st century, we've got some additional tools at our disposal to help with these two tasks as we move forward and try and synthesize new and improved varieties. So in the department of creating new useful genetic variation, we have now biotechnology at our disposal, and also some statistical techniques that I'll broadly label prediction. Mainly these are focused on getting estimates of breeding value in potential parents, mainly through the use of molecular markers. Then on the side of identifying outstanding progeny, we've got some tools like molecular markers that can be used in a broad category of tools called genomic-assisted selection. So we're using their DNA-based or RNA-based or protein-based information along with phenotype to help to identify what those outstanding progeny are. We can utilize phenomics, which is very generally defined as the interpretation of phenotype, evaluation and interpretation of phenotype in a particular environment. So normally breeders are evaluating the performance of progeny in a very broad range of environments that represent the target region that they're breeding for and trying to develop a new cultivar for. And phenomics is much more high throughput, often automated, and often uses specialized testing environments instead of just the environments that represent the region targeted for the new cultivar. So an example is breeding for drought tolerance. You might have a specialized testing environment that's very high management to specifically look at a certain tolerance of the plant for growing and yielding well with certain amounts of moisture input. And so all of these tools are helping us a great deal in our efforts to develop new and improved varieties. And then as we learn more about genetic architecture and gene function, that information is again leading to better use of genetic variation in the ways that we put that together and then exploit it. So this is a cycle that plant breeders will repeat many, many times, hopefully in their career. Advancements that are made in developing new cultivars then generally utilize those materials along with others to when they start the cycle again of creating that useful variation. So in other words, advancements build on each other. So we'll talk more about specifically where biotechnology fits into this overall picture. I think of biotechnology in terms of transgenic traits. So these are novel traits that are accessed through transformation. Previously, breeders could only get access to genetic variation in terms of sources that would cross to the target species. What transformation does is it virtually opens the gene pool to access traits from any species, basically. In addition, the genes from the target species can be modified to produce new forms of trait expression. For example, expression in specific tissues at higher thresholds, and even engineered to be silent, so not express themselves. An event is a source of a transgenic trait that's produced through transformation, and it's defined in terms of the unique DNA sequence that's inserted itself in the target genome through transformation and the precise point of insertion. So it's important to remember that transgenic events that go out into the commercial arena and in the marketplace have each originated as a single plant. We refer to as a T0 plant. And this is the product of being regenerated from a single transformed cell, as John Feiner showed us yesterday. So this schematic shows the two avenues of transformation and how they result in that single plant. A transformation vector is created. It includes, at minimum, the gene of interest, a promoter for that gene, and a terminator sequence. And it can be introduced to the target species either using agrobacterium as a means of transport and introduction, or through bombardment and use of what we call the gene gun. In either case, the DNA is introduced into the target plant genome. Cells that have been transformed are screened and isolated from others, and then those transformed cells are regenerated into a plant that is fertile. Fertile is very important because without that plant being fertile, then a seed crosses can't be made, and that event can't be moved into other materials. There's been a very rapid rate of GM crop adoption in the U.S., and this graph shows the rapid rate of adoption with GM corn. So last year, 88% of the corn crop in the U.S. Included a transgenic trait, and 49% of the corn grown in the U.S. included more than one transgenic event. The types of traits that have made it to the marketplace or are in development are covered partially by this list. This is not necessarily all-inclusive, but it's meant to show that there's a diversity of traits. There's some that protect the genetic potential of the hybrid that's developed, and I put herbicide resistance, insect resistance, and stress tolerance into that category. So they're traits that are actually preventing pests or environmental situations from robbing that genetic potential that plant breeders have put into the hybrid. And then there are transgenic traits that actually extend that genetic potential. Their yield and productivity enhancements, and possibly enhancements on the nutritional side. Well, the development of biotech traits, by and large, happens in the private sector. This is a slide that they found on the internet that shows Monsanto's pipeline. I think it's a good representation of a general pipeline that would be involved in transgenic product development. So from gene discovery through market launch, there's basically 4 phases involved. First phase is identifying elements that would go into a commercial vector. Phase 2 is creating many, many events and beginning to evaluate them. Phase 3 is basically focused on testing of events and then beginning to introduce those events into elite commercial materials. And then phase 4 is where the regulatory package is submitted to the government. It includes very broad trade integration to move the single commercial event into a very broad range of genetic backgrounds. And then those hybrids that are converted are subjected to performance testing in a very wide range of environments. All of this is happening at the same time that seed is being bulked up for commercial seed volumes before market launch. So what you see through this process is that very, very many events are created and then subjected to intense evaluation and very intense selection to identify a single event that will be finalized for commercial approvals and then brought to the marketplace. So there's many events that are created that are only put in the field under containment practices, so they're not released into the environment, it's basically only the event that is approved by the government that is grown on a wide scale. Event selection criteria might include a list something like this. Single copy, structural fidelity, and by that I mean great similarity to the DNA that was introduced through transformation or intended to be introduced. Desired expression level. So is the event expressing at the proper threshold, in the right tissues, at the right timing in plant development? Is there consistent expression of the event in terms of a certain threshold? Is that threshold consistently expressed regardless of the environmental conditions? For example, the stresses that that plant might be under, the genetic background that the event is moved into, because obviously it would be desirable to move the event into a very wide array of the elite hybrid lineup in corn, for example. And then in addition, is that event expressed at the threshold levels even in the presence of other events? Because as we've seen, there's definitely a trend to start stacking events and to offer more than one event in an elite hybrid background. So you want to make sure that the events are compatible with each other. Preferably, we'd like this consistent, desirable level of expression with only one copy of the event. And so you can imagine in corn, where hybrids are the cultivars, we'd like to be able to convert only one side of the pedigree, not having to convert both sides. That's a convenience factor, and not a requirement strictly. We'd like stable inheritance over generations. And finally, we'd like to ensure the absence of negative effects. So as plant breeders, we look for potential problems in several areas. For example, is that structural fidelity there? Is there gene silencing potential, especially under stress or with the presence of duplicate DNA sequences? And one example of that might be if there are two separate events that have, for example, the same promoter. There's the opportunity for one event to silence the other. Is there any interaction with native genes? So as we've seen in corn with herbicide and insect resistance, basically the plant is producing a novel standalone protein in the plant. There aren't metabolic pathways that are involved, for example, with producing Bt protein except in a transgenic crop. And so there's generally not that propensity for the Bt gene to interact with other genes in the genome. But this may change as we get into traits like drought tolerance, for example, or we begin to change grain composition, and so we're actually there's some possibility there that the transgene would interact with other genes in the genome. So we watch for it. And then finally, we want to ensure that the performance for other key traits in the plant aren't negatively impacted. So, for example, one thing I could envision is that, say, the event has inserted itself in the corn genome right smack on top of a very important gene for yield, in which case the insertion site might interfere with yield performance. So these are the kinds of things that we screen for as we're evaluating the various event candidates. So what does event testing look like? It starts with molecular analysis. So we're looking for that single copy that shows the structural fidelity of the DNA for introduction to the target species. And then efficacy testing basically is focused on is there proper expression for the trait of interest. And this is done with very high precision experimental designs. So one example of that would be a replicated split plot design that uses isogenic lines. So the hybrid with the trait versus the hybrid without the trait, and in the presence of very intense trait pressure. So if that's an insect resistance gene, there might actually even be infestation, artificial infestation of the plots with a really high level of insect pressure to see what the upper limit is for insect resistance among those events. Then there's yield performance testing, and this would be done both in the presence and outside the presence of the trait pressure. And then there might be other types of tests to actually evaluate the impact. So for example, if it's a nutritional trait, there might be feeding studies if it's intended as a nutritional enhancement for, say, swine feeding. Testing environments would include a very wide representation of environments in the target market region, for example, covering soil types, covering differences in farmers' cultural practices that are employed, say tillage practices. Would happen over multiple years to catch different climatic snapshots and under a variety of stress factors. And I'd like to emphasize this because stress is an important part of the testing because it's under stress that some events can show silencing. A variety of genetic materials would be used to make sure that it's compatible in many different environmental genetic backgrounds, and then stacks with different combinations of events. So the ideal event for commercialization is one that results in dependable trait delivery, no extra baggage, and a practical utility in that the event, in the ideal sense, acts with dominant gene action. So you only need one copy in a hybrid. And I'd like to just mention, too, that there's a trend in transgenic product development toward including multiple modes of action for a particular transgenic trait. And so I use this slide of SmartStack that was released by Monsanto and Dow back in 2007. So it's a little old. It's saying that some of the regulatory reviews are still pending, which of course that's not the case. But what this does show is that there's 3 genes for above ground resistance, 3 genes for corn rootworm below ground resistance, 2 different modes of action for herbicide tolerance. And this basically lends more durability to the transgenic trait that's being offered. So as a plant breeder, I know that whenever I'm working with a single gene trait, whether that's transgenic or non-transgenic, I know that it's just a matter of time before nature overcomes that gene. So I like to work with genes, traits that are controlled by multiple genes, because with every additional gene, there's an exponential reduced risk of resistance being developed in counter. So this is a movement that's also going on toward multiple modes of action. Okay, I'd like to switch now to the trait introgression pipeline. And with corn, basically we, as an industry, commonly practice backcross breeding as a means to introduce the transgenic trait into an elite background. So what this represents is an intersection between the biotech trait development pipeline and the pipeline to develop new and improved hybrids in the case of corn. So the goal with backcross breeding is to get back, recover all the attributes of the elite hybrid that you've targeted for conversion, plus that extra value-added trait as kind of frosting on the cake. And it involves a number of generations. Basically, backcross breeding would start with a cross to a source of the transgenic event. We call it a donor. And the recurrent parent would be the line targeted for conversion. So you see here 7 crosses to the recurrent parent. And typically this would be required to get back 99% of the genome of the recurrent parent. Then there's 3 generations of selfing to get that event to homozygosity and fix that trait. And then there's 1 generation in corn to actually produce a test cross, because the converted hybrid will be subject to very wide multi-location performance testing. So a little backcross theory. With every cross, we're getting more and more of the recurrent parent genotype back again. So with the first cross, we get 50% like the recurrent parent, but with the 7th cross, we're up at 99%. Basically, with every cross, the DNA that's not like the recurrent parent is being reduced by half with every generation. It's important to remember that each backcross generation has some variability in it. So even though, you know, a backcross 1 generation is on average 75% like the recurrent parent, the individuals in that population actually differ for the real amount of recurrent parent germplasm that's been recovered, and where those chromosomal segments that are not like the recurrent parent are located in that genome. So one thing that breeders do is we use molecular markers as a tool to help us to speed this process along and to identify those individuals, for example, in the upper tail, that have more recovery of the recurrent parent. So for example, with this distribution, you can see that that upper tail would include individuals that have 85% or more, like the recurrent parent, quite a bit higher than the mean for that population. So we can reduce the number of generations that are required for this process quite significantly by utilizing molecular markers as a tool in this process. So we accelerate the conversion table by using markers, speed time to market, and we can also do something about selecting against DNA that's not like the recurrent parent in the immediate region flanking the event. We call that linkage drag when there's DNA that comes along with the event. It doesn't necessarily represent more risk it's just extremely stubborn and difficult to remove without molecular markers. So markers are a great aid in that. And lastly, markers can be used to save a generation or two because they can be used to help identify event homozygotes in the selfing process. So markers can be used to produce a more timely launch, get transgenic traits to market sooner, in elite backgrounds, and in a more efficient manner. And then finally, I just want to touch on yield testing a tad, because those converted hybrids are subject to very high scrutiny to make sure that all the attributes of the original elite hybrid have been recovered along with the presence of the new value-added traits represented by the event. And the success rate in achieving what we call equivalent performance, not to be confused with equivalency that we're talking about in other respects, but this is equivalent performance mainly on the yield level, and that success rate is a function of the amount of the non-recurrent parent germplasm that's been retained in the backcrossing process, and the number of versions of each conversion. So I've only talked about one version being produced, but actually many organizations commonly produce more than one version of each conversion to make sure that when they get to the end of the process, they actually have a product to sell. So there's something with equivalent performance plus that frosting on the cake that's commercializable. Then finally, seed volumes that are produced for commercial sale are quality controlled monitored to ensure that event identity and purity are as consistent with the labeling on the bag. This is to confirm the presence of all intended events and at the specified purity levels. So for example, a bag of seed might say 98% of the seed in this bag carries this trait, and so this testing is done to ensure consistency with that. It's also done to confirm that there's the absence of any unintended events, and we call this adventitious presence. So we want only the events that are desired, and we want those at the specified purity level or above. And this testing is done in addition to other testing that's done on any hybrid seed to confirm the line and hybrid purity, uniformity, seed viability, and seed quality. And then finally, quality control monitoring is backed up by quality assurance, which is involved with standard operating procedures representing best practices for the activities in the pipeline all the way through. And this really ensures not only quality but uniformity across the entire organization and the pipelines involved. So in conclusion, biotechnology provides a key advantage in crop improvement because it allows us to tap into useful genetic diversity that's not otherwise accessible to the plant breeder for the target species. Transgenic events are subject to intense evaluation and very stringent selection prior to commercialization. Commercial candidates are introduced into elite cultivars, often using molecular markers, to offer farmers the biotech traits in desirable agronomic packages. So we're interested in creating a whole agronomic package that's of value to the farmer. Final seed products are performance tested and quality inspected to ensure genetic and event identity and purity. And I think it's obvious that, hopefully it's obvious, that GM cultivars are subject to a significant amount of testing, even more testing than typical products that would go to market using conventional plant breeding practices alone. Thanks so much for your attention.
Hi, good morning. Owen Hookinga, USDA. I'm wondering if you'd like to comment on the use of zinc finger nuclease technology to do DNA editing and resequencing without having the baggage of integration events from outside the genome.
Thank you, Owen. Okay, so you're speaking of a technology that would be, sure. So the question was asked about the use of zinc finger technology, and I'm going to include that, if you don't mind, in a broad category of technologies that would direct insertion of introduced DNA to a specific site. So what I had mentioned in my presentation was more random insertion, which most of the commercialized events that are out there now are a product of. But there are technologies now to direct insertion of DNA to be introduced through transformation to certain places in the genome. And so this is a great example of, as we know more about genetic architecture and genome function, if we can direct introduce DNA to specific sites that we know reduce the risk of unintended effects. So I use the example of an insertion site landing on an important gene for yield. Obviously, we would eliminate those risks if we could direct insertion to sites that we knew were going to result in viable and consistent and stable expression of that DNA. Yeah, it could be a huge help.